Chapter 3 looks at the cell and the parts of the cell. We didn't know about cells until the microscope was invented. Our cells are quite small, require a microscope to be seen. Once they discovered the cell, they started to look at all kinds of living things and realized that cells make up all plants and animals. Some more observations led to the cell theory. In science, a cell theory is as strong a statement as you can make because all evidence from any experiment supports that theory. So the cell theory states that all living things are made of cells, all new cells come from pre-existing cells, and the cell is the smallest unit the smallest unit of life that performs living functions. So our cells arose from a single fertilized egg. Once fertilized, it became a zygote, and the zygote started to divide. At first, all of these cells are identical, but changes occur throughout that division process that caused cells to differentiate or specialize into different types of cells. Each cell is a living thing that must maintain homeostasis. And this is talking about a very interesting process of cell division of the fertilized ovum, that very first zygote, that first cell that was you. That first original cell division is called cleavage, and with each cell division, the cells become smaller and smaller and smaller. And as these cells divide, the DNA is affected, and the DNA is turned off in some cells, turned on in other cells, and it creates differences in the cells called differentiation. Once all of the cells have differentiated, there are basically 4 kinds of tissue. Tissues are made up of cells that have the same structure and function. Here is an example of cells dividing. This was taken with an electron microscope, specifically a scanning electron microscope that allows you to see a 3-dimensional view of the object. Our cells are surrounded by fluid. They're basically in salt water. So the fluid that surrounds our cell is salty water. This fluid is outside the cell, so it's called extracellular. But the fluid is usually called interstitial fluid. That is the fluid that is in tissues. Cells have basic components. Every cell has a plasma membrane or cell membrane, and that separates the inside of the cell from the outside extracellular fluid. Cells have cytoplasm. It's the material between the cell membrane and the nuclear membrane. It's a colloid, meaning the proteins are dissolved in it and they won't separate out. Jello is a colloid. There are 2 subdivisions of the cytoplasm. The cytosol is the intracellular fluid, and the organelles are the little organs or individual structures that have very specific functions. Organelles may be non-membranous. That means they're not enclosed by a membrane. Some examples are cytoskeleton. This picture here on the right shows the cytoskeleton and all the proteins within the cell. It's stained in a special way so you can see the proteins that maintain the shape of that cell. Microvilli, we'll talk about those later. Centrioles, all these objects we'll talk about later. Cilia and ribosomes are non-membranous. Membranous organelles are enclosed in a membrane. The membrane is just like the membrane that encloses the cell. It's made of phospholipids. Examples of membranous organelles: mitochondria, the nucleus, endoplasmic reticulum or ER, Golgi Apparatus, lysosomes, and peroxisomes. These are all of the membranous organelles that we're going to talk about in this chapter. This picture here is of a plant cell because these are chloroplasts that are enclosed in a membrane. A plant cell might have hundreds to thousands of chloroplasts. So we're going to talk about the individual organelles, structure and function. Some cells have So if you look at the upper left, you're going to see the cell membrane here, but some cells have these extensions of the membrane, and this will increase surface area. This is called microvilli. We'll see this in the small intestine. Over here on the right is another example of the microvilli. These extensions increase the area for absorption in the small intestine. The cytoskeleton is made up of protein filaments. The centrosome is an organizing center for cell division, and the centrosome contains two objects called centrioles. Now here are the centrioles. They're made up of they're really similar to a type of protein called a microtubule. They organize the microtubules, but it's this object where these cylindrical structures are at right angles to each other within the cell. For cell division, these divide and migrate to opposite ends of the cell to organize protein fibers so that the chromosomes can move during cell division. Ribosomes are made in the nucleus inside the nucleolus. A ribosome is made of RNA, ribosomal RNA or rRNA, and proteins. You're going to find ribosomes attached to the ER. In this case, the ER is called rough ER, or rough endoplasmic reticulum, and you might find the ribosomes free in the cytoplasm. If the ribosomes are attached to the ER, it's making proteins for export from the cell. If they're free in the cytoplasm, it's making proteins to be used in the cell. Organelles, membranous organelles, include peroxisomes. It's a small vesicle that contains enzymes to break down organic compounds and to neutralize toxins. Lysosomes, if you looked at a cell, you wouldn't be able to distinguish a lysosome from a peroxisome. It is just a little vesicle. But in the case of the lysosome, its function is to remove damaged organelles or pathogens if the cell is a white blood cell and engulfs a bacterial cell. The lysosomes will come over and fuse with that bacteria and digest it. Organelle, such as the Golgi apparatus, is involved in the alteration and packaging of proteins for export, and it looks like flattened pancakes. You'll find it near the cell membrane. Mitochondria has a double membrane system. The outer membrane goes around the outside. The inner membrane folds to form what are called cristae. And the function of the mitochondria, it's often called the powerhouse of the cell because it uses our nutrients such as glucose to make ATP. The nucleus will be the largest structure in the cell. It contains the DNA, enzymes, and proteins. It, like the mitochondria, has a double membrane. The endoplasmic reticulum is associated with the nucleus, but it's a network of interconnected tubules, and on the outside of the endoplasmic reticulum will be ribosomes if it is rough ER. If the ribosomes are the protein synthesizing factories, and the protein that's made will enter the rough ER for transport and modification. If it's smooth ER with no ribosomes, that function is completely different. The function is to synthesize lipids and carbs. So these different components of the cell function together for the manufacture and export of protein. So there is a pathway for proteins to be produced in the cell. They are produced on the exterior surface of the rough ER. The protein will enter the rough ER for modification. It will be packaged in a little vesicle. That little vesicle will squeeze off and travel to the Golgi apparatus. Possibly more modification here, concentration into vesicles, larger vesicles, that then transfer over to the cell membrane and fuse with the cell cell membrane to export that product. A good example would be a cell in the pancreas producing insulin for export. The smooth ER has different function, making lipids and carbohydrates. Here you can see a lysosome. Now the lysosomes are made within the cell. So here we have the Golgi apparatus with digestive enzymes in it, squeezing off to form this lysosome. If a bacterial cell were to come into the cell, it would be surrounded by its own membrane. This lysosome membrane can fuse with the bacterial membrane, release the digestive enzymes, and digest the bacteria without damaging the cell. Which of the following organelles is often associated with ribosomes? Rough ER, for the production of proteins for export. Which of the following organelles is not associated with the modification, packaging, and secretion of proteins? Smooth endoplasmic reticulum makes carbohydrates and proteins. In what organelle are steroids, triglycerides, and phospholipids synthesized? Those are all lipids, so they're produced in the smooth ER, or SER. Which of the following organelles is not involved in the production and flow of proteins? The mitochondria are involved in the production of ATP, cellular respiration. Which of the following structures is ribosomal RNA synthesized? The nucleolus, which is inside the nucleus.